So, good morning, everybody. So we obtained two data sets from an ex vivo Marmoset brain. The first one is, so we segmented many subcortical areas using multimodal MRI and the histology. Finally, we generated a three-dimensional digital template atlas from these segmented subcortical areas, also called SAM. It's also called Subcortical Atlas of the Marmoset monkey. So there's nothing to declare here. So why Subcortical Atlas? A comprehensive three-dimensional digital atlas of subcortical region in non-human primates based on MRI and histology is of great value in anatomical, functional, and connectional studies. In particular, a 3D atlas can be registered to a non-human primate brain MRI volume to best determine the region of interest for anatomical tracer injections for connectome studies, the areal location of observed functional activation areas in fMRI, and the potential target for the non-human primate model of the neurological disorders. Many of these subcortical areas, the nuclei and their subregions are challenging to identify and delineate in conventional MRI like T1 or T2 due to their small size, hidden location, and often subtle contrast compared to neighboring regions. To address these limitations, so we combine high-dimensional MRI, including the mean apparent propagator MRI, and also called MAP MRI, along with the 9 microstructural parameters, along with the T2 weighted and the MTR images, and the matched histological sections with multiple, in this case, like 5 different strains derived from the same Marmoset brain specimen to delineate subcortical regions and associated white matter pathways for 3D digital template atlas of the Marmoset in ex vivo. So we scanned a perfusion fixed adult Marmoset specimen and a 70 horizontal bore magnet using MAP MRI and other MRI parameters with 150 micron resolution. Before the MRI, a 3D mold was fabricated using 3D printing to align the specimen in stereotaxic coordinates. The brain was positioned in the 3D brain mold and inside a cylindrical tube filled with the formalin. Subsequently, the tube was sealed and placed inside the 30 millimeter quadrature millipede coil for MRI imaging. We computed these microstructural DTI MAP MRI parameters. These are propagator, fractional anisotrophies, mean axial and radial diffusivities, non-Gaussianity, return to origin, axis, and plane probabilities, along with the fiber orientation, distribution, or FOD. In addition, we also acquired MT and T2W scans with 150 micron resolution. The scanning parameters are described in details, so in these references here. Following the MRI, the whole brain specimen was prepared for histological processing with multiple stains. We derived MRI and histology information from the same brain specimen. So we cut the serial 50 micron frozen section through the entire brain specimen from frontal to the occipital, the cortex, and stained the adjacent series of sections with parvalbumin, SMI32, acetylcholine esterase, chrysal violet, and choline acetyl transferase. So the high-resolution images of all the histology stained sections scanned using the wide field microscope and the slide scanner. So we then matched the corresponding histology images with the MRI images from the same brain specimen to identify and confirm the brain areas. It is important to emphasize that we did not rotate the brain specimen to match the histology section. Note the correspondence of the deep brain structures in both MRI and histology. Next, we segmented these areas for 3D atlas with reference to MRI and histology. Let me expand this part in the next couple of slides here. So we used a combination of these different MRI parameters to distinguish the architectonic borders of subcortical areas from the neighboring structures. So the deep brain region show the distinct and a distinct contrast in different MRI parameters. You can see the thalamic structures, the contrast is different in different MRI parameters. For example, if you can see the subregions of the globus pallidus is well defined in the MTR compared to the deck map. But anterior thalamic nuclei and the hypothalamus are well defined in both MTR and the Deckford images. We then manually traced these areas through the 174 serial MRI sections for the 3D atlas using matched histology sections and the multiple stains as a ground truth. So this example shows the different subcortical nuclei and fiber tracts in the Deckford on the left, Deckford map MRI images, and the matched corresponding histology section stained with the SMI32 and the chat for 3D segmentation. These coronal slices were arranged from this is the rosal and this is the caudal. We then converted the 2D segmented subcortical areas into and the deep brains into the 3D dimensional volume. We adopted this new ex vivo 3D volume as a SAM, also called a subcortical atlas of the Marmoset monkey. We made the symmetric version of this atlas. So we integrated the symmetric atlas into the AFNI and the SUMA software packages with the subcortical area labels. So this illustrates the new symmetrized version of the SAM atlas with the segmented deep brain regions in the coronal, horizontal, and the sagittal plane of sections. We mapped 251 brain subcortical areas, including subregions of the cerebellar cortex and the hippocampal formation. The illustration at the bottom shows the spatial location of the delineated the subcortical areas on the dorsal and the lateral views of the brain, lateral views in 3D. The new template atlas is intended for use as a reference standard for Marmoset neuroanatomical, functional, and connectional imaging studies involving subcortical target. The digital atlas is now available in the AFNI and SUMA analysis package and can be subsequently registered to 3D anatomical scans of other individual Marmoset control adults to guide the research applications that require accurate knowledge of the areal boundaries, as shown in the next two studies. In this example, the ex vivo SAM atlas is registered to an in vivo T2-weighted population-based standard template, also called the MBM template, oriented to the year bar 0 stereotaxic coordinates. The illustration in the D and the E shows the registered deep brain regions on the T2-weighted control template. So none of these registered regions were altered or adjusted. The results show that the spatial location of transformed segmented areas in the T2 averaged volume corresponds well with the delineated areas in the SAM atlas. In the next example, so in another example, the ex vivo SAM were registered to 6 in vivo control adults of different age groups here. So ranging from 1 year, the C, and the 10 years, the H. The result shows that the corresponding location of the registered subcortical regions in the control individuals matched well with the SAM atlas on the top left. They also demonstrate that the straightforward affine and nonlinear warping are sufficient to distinguish and to provide atlas-based estimate of the areal boundaries in the Marmoset subject in vivo. To summarize, using combined multimodal MRI and histology, we segmented 251 subcortical deep brain regions, including the cerebellum and hippocampal formation, and generated a 3D digital template atlas, also called SAM. So the strength of the work is this study utilizes the high resolution MRI, 150 micron resolution, and the corresponding histology with multiple stains from the same brain, also called MRI histology correlation. This integrated multimodal approach yields more objective and reproducible delineation of the nuclei and their boundaries in deep brain structures. Finally, we estimated and confirmed the atlas-based aerial boundaries of the deep brain regions by registering this ex vivo SAM into in vivo T1 or T2 MRI data sets of different age groups. So, conclusion that tracing and validating these important deep brain structures in 3D will improve. There are several techniques we can apply to this one. So, the neurosurgical planning, anatomical tract tracer injections, and navigation of the deep brain stimulation probes, and the functional MRI and the brain connectivity studies, and our understanding of brain structure function relationship. So we also generated the macaque atlas, also called D99. So the difference between the macaque and the marmoset is the macaque includes both cortical and the subcortical atlas. But in the marmoset, it's only subcortical, just we are working on the, so now it's a cortical version of the marmoset. It will be complete. So finally, I thank all my collaborators. So they're involved in a different part of the work, actually, all like, the MRI, histology, and everything, okay? So I'm happy to take any questions. Thank you very much.
That was lovely, thank you. If there's questions, you can put them in the app, or if you're brave enough, you can also come up to the microphone. So we already, well, there we go.
One question about the histological stains. It would seem like it would have been an opportunity to include a myelin stain that you could have compared with the magnetization transfer. I was wondering, did you guys think about that?
Yes, we have from the different data sets, but not from the same monkey. But we wanted to use like some kind of antibodies against the myelin, not the regular histochemistry, but somehow it didn't work out actually. So that's why we use like a completely, we have the completely different data set, but we didn't include into the, if we need, we can work on that one too. So now we are also working on the fiber tracks too. We're going to use the myelin data set.
Lovely, there's a few more questions for you. So first one is, wonderful resource. What is the spatial resolution of the histological data?
50 micron. No, spatial resolution means, so what do you mean for histological data? We just cut like 50 micron thick section.
I guess that answers the question.
Maybe they mean, do the digitized images of your histological section, is the in-plane resolution high enough that you could do visual microscopy? So one pixel in your image is equivalent to how many microns in the section?
Oh, okay. That part I don't know yet, but we can zoom in to see all the details in histology.
So you can really see single cells in this.
Yes,
Okay,
yes,
right. Then it's probably.
So that means each section is like 36 gigabytes. That means we can zoom in to see the details, all the details.
Okay, two more questions for you. At what age do marmosets reach the adult size?
Oh, okay. So even if it's 1 year old, so it looks similar to 10 years old. So maybe I think by birth, actually, some of the brain structure looks similar to the adults, I can say. But I don't know exactly when it is, the brain structures are mature. So probably, I don't know the developmental studies about, but we compare like 1 year versus 10 years. The prefrontal and the other structures look similar to me. In MRI, we compare like a different MRI, like T1 and T2, they look similar. But I don't know about the histology. So we have the histology from 10 years old.
And the final question is, are nomenclatures of labeled subcortical areas in marmoset identical to those in humans? Any differences among species?
Very different as compared to the human, of course. So if you look at the prefrontal, and the people have mapped the prefrontal cortex of the marmoset with macaque. So we can see that because it's, if you look at the sulci and all, don't compare the human, but compare the marmoset and the macaque. We have just like what's called the lateral sulcus. If you want to map out what's called the area 8A, it's not very easy in marmoset. So compared to like macaque. Macaque, we have the arcuate sulcus and the principal sulcus. But in the human, it's very different.
Lovely, thank you.